tell you the story of this pretty pink yeast that I found in the environment and managed to change its color. So I had this weird stuff growing in some hydroponic nutrient, and I actually managed to send some off for sampling and figure out that it was a whole thriving community of different microbes. And you can see there's actually even some algae growing and a whole bunch of different bacteria and some other things as well, little individual algae cells. So I took a swab of it on some agar plate just made with Marmite and some beef broth and agar and water. And all sorts grew, including this tiny little pink colony, which I thought I'd grow out. Looking at it under a scope, you can see it's these sort of large, or relatively large compared to like bacteria cells. They stained with methylene blue, and some of them stained a lot more than the others. This is consistent with yeast. When they're dead, they stain a lot darker. So you can see the sort of clumps are often the dead ones come together, and then the sort of less stained ones around the edge are living and thriving. So I streaked it out a few different times trying to isolate just the pink colonies. One of the attempts got contaminated with this gorgeous yellow, presumably Micrococcus or some other sort of bacteria growing in there in around the yeast. But I managed to get back to some pure yeast colonies. And so then the next thing or the final thing I guess that I wanted to try with it, other than I doodled some around in some agar art, but I recently took delivery of a 250 nanometer UVC germicidal lamp. So it's not something to play with as a toy. They can be quite bad for your eyes and skin. So I keep mine. I wear safety goggles at all times, and it only is ever on inside a sort of light, tight box. But I thought it would be fun to try irradiating this yeast, and using that staining thing, you can figure out how long it takes to kill most of them, and so leaving only a few percent alive, irradiating it for, in this case, 4 minutes, just to see if we could induce some mutations. And so obviously most mutations are not going to be visible to the naked eye, but the color in these yeasts comes from various carotenoids. So I thought if we could have some mutation that messed with any of those mechanisms, we should see a visible color difference. And we actually got a few colonies that indeed have substantially less or potentially even no carotenoid production. So something in that pathway, some gene that's involved in making those colors, might have been knocked out with the mutation incited by the light. I did also try, let's see, other pieces was some of the times I added caffeine, which can be sort of help cause mutations to stick around in some cases. And I also made sure to put it in the dark soon after the exposure to the UV so that it doesn't have time to repair. I still found it surprising, like 4 minutes of exposure to this lamp. This is way more than most things can take. And actually, this yeast in general has been a bit of a pain just because of how hardy it is. I was also trying to crack it and extract out the carotenoid pigments for some other experiments, but dang, this yeast is a surprisingly tough nut to crack. And even with 4 minutes of exposure under a germicidal lamp, plenty of the cells, like too many of the cells to survive for me to get nice isolated colonies, and only a couple showed this visible mutation. So anyway, that was my fun experiments with this environmental yeast, just picked up from something growing in an arbitrary cup in my basement. I hope you found that interesting. Thanks for watching, and we'll see you in the next one.